at the site F508. As a result, this mutant protein does not fold properly. It's retaining the ER, and it will be degraded eventually. So in this assay, we're looking for small molecules or compounds that can promote this mutant protein trafficking to the cell surface. So for this purpose, we have generated a Fab CFTR delta F508 construct where we added this transmembrane domain between the Fab tag and the CFTR N-terminus. So after expression, Fab will be expressed on the plasma membrane, and it will be in the extracellular environment where we can add the extracellular dye to stain this particular pool of proteins at the plasma membrane. And then here are some confocal images showing that with the extracellular dye, you see there's like very small amount of protein on the cell surface. Basically, there's like no signal. However, if you use the S dye, which can get into the cells, now you can see there's a lot of fluorescent signal coming from within the cell. And after we treat the cells with the compounds that can rescue this mutant trafficking, now you see there's now a lot of proteins at the cell surface. We have created a similar vector where we basically just replace Fab with GFP. And in this case, you can see that there's GFP fluorescence throughout the cell, and it's just impossible to detect the cell surface pool of the protein of interest. And for running the assay, we use flow cytometry as our readout. And in this graph, you can see we have the untreated population, or the cells treated with just DMSO, and these are the cells treated with the corrector compounds. You see the shift of the mean fluorescence intensity. And we have also combined corrector treatment with low temperature rescue. So in this case, the cells were treated with a combo of correctors C4 and C18 for 24 hours. And also the cells were incubated at 27 degrees. So from this, you can see there's a huge increase of the cell surface pool after this treatment. The same thing happens when you use the S dye to label the total protein. You see this big increase of the total protein as well. And then we get a Z prime greater than 0.7, so that means we have a really robust response under this condition. So in summary, the Fab-based assays could be used for high throughput screening, or it can be used for multiplexing purposes. You can use it with your typical fluorescence readout, as long as it's a different color, and you can use it with your luminescence readout. So you can use for your primary assays or any follow-up assays where you want to study more detailed live cell imaging. And also, we have the fluorogen dyes. They are available for purchase. So you can be used for compound profiling or a full screen. We have all of the cell lines available. And in addition to the CFTR cell lines I just talked to you about, we also have the PGP cell line. So you can use that to see if your CFTR compounds are very selective or not. We provide assay services. So if you have any target in your mind, if you want to do protein trafficking, please come talk to us. And I'm also having a poster, and my poster is number 43, I believe. And with that, I would like to thank my boss, Dr. Scott Snedden. He's the president and CEO of our company founders, Dr. Marcel Boucher, Dr. Alan Wagner, and everyone working in the lab, scientists. Thank you very much for your attention.
Thank you.